A very big hello to all my wonderful PU2 champions. I hope my dear friends are doing absolutely great. And now you might be thinking, ma'am, how do you think we'll do great? As of now, we do not have much time for biology. So listen to me carefully. There are two people you have to put your trust on. Okay, ma'am, you right? You're talking about yourself. I will come into the picture. The first thing that I want to tell you, you have to keep a trust on yourself. Suppose your name is Gia and you are thinking that I will not be able to do biology. I have so much left. There are so many 13 chapters and this is a theory subject. How will I remember all of these questions? Now questions are coming from anywhere also. I will not be able to do it. So Gia or anyone who is listening to me, listen to me very, very carefully. You can make your mind in a different way and just give yourself a chance by trusting yourself. Think that, okay, this is very difficult time for me. Probably I just have one day, but I'll try to make the best use of it. I just try to make the best use of it. I have the trust on myself that if I try, I might get at least something really, really good. And that is exactly I want from all of you. First person that you have to trust is yourself. The second person you have to trust is me. But without trusting yourself, if you trust me, it's of no use. You get it? So you have to work really hard. I am here with you. I will be helping you with each and everything. Now, those of you, I want to tell you something straightforward as a teacher, as a genuine person out there. If you feel that in biology you have not touched anything, I do not know anything. There's a video that I did, okay, where we merged all the 1 marker very important questions, 2, 3 marker and 5 marker questions. There we merged it and we kept it. It's around 10 hours long. It was stimulated as live. It was a recorded video, but it was stimulated live. 10 hour is an important factor over here because I know you are very concerned about the time. Only if you feel that I have no chance for completing the entire syllabus or do not know what to do, this is one video that you can refer to. It just came into my mind. You can refer to it. We had published it before. If you want me, just ask the link. I'll put it down in the comment section. Yes, if you want the links, or not want me, if you want the link. That is one way of dealing with things. Now, the second way is that you are someone who is, basically you have covered some chapters here and there and you just want to be pro. You want to really score 70 on 70. That is your target. Then make sure you practice all the questions that are given over here. Again, the 2, 3 marker that I have solved with you is there. The 5 marker that I have solved with you is there. But there are a few extra questions as well which you have to solve. And this gives you an idea on where should be your focus going on. Okay, so starting with the first chapter, that is sexual reproduction of flowering plants. I told you before also, there is a way to put all the important topics like this. But I do not prefer it. It looks very easy and things like that. Children get attracted to the easy stuff. I am really against it. Listen, you should know how the questions are going to be framed, right? Yes, that is the motto over here. So the 5 marker questions over here could be framed like this. The topics are the same only. There are different ways the same question can be asked. For example, you might have seen a question in your question bank about hexaploid condition and things like that. You might have never seen that. So please know these type of questions could be coming in. Practice each and every them. I have made a video on it separately. If you're someone who wants to do it right now, go watch those videos also. 2, 3 marker, 5 marker videos that I've done. You can just skip it and look for the chapter wise segregation I have also done. Make sure you watch it. From this chapter, double fertilization related questions can come. Gametophyte as in female and male gametophyte and outbreeding devices have more chances to come. These are the questions that have, these are the concepts which have more chances to come. And when I say more chances to come, I cannot be saying that exact question definitely will be coming in. I am a teacher. I have not set the PU board paper, right? So yes, I have studied MQP, I have studied the question bank, and with that I'm telling you, please do not miss out on these questions for sure. Okay, these are 5 marker. Apart from it, you will also have the 2 and 3 marker questions. Again, please understand in 2 marker, you have pollen grain, true and false fruits, seed related concepts that have more chances to come. Pollen grain as in the diagram of pollen grain can be coming in, or probably you can get sporopollen related questions, or probably the cells of the pollen grain out there, the nucleus of the pollen grain cells. Okay, the true and false fruits, you should know apple and cashew are the examples. What are the parts that forms all those fruits? You should know about that also. Seed related concepts, albuminous seeds, non-albuminous seeds. Can dicots be albuminous or non-albuminous? Know all the basic things about these concepts are there. Questions are not going to be repeated, concepts are. Okay, concepts are. So if your concepts is strong, different form of questions, you will be able to do it. These are the questions that have probable chance of coming up, but yes. Apart from it also, I've given you certain other questions which you have to practice. Now in 3 marker, definitely there are more chances, super more chances for what? For diagram based question to come from this particular chapter in 3 marker category. Make sure you're pro in this particular diagrams are there. Apart from it, I've given you one extra question also which you have to solve. Cool? Yes. Now, next, human reproduction, you have 3 marker questions coming from this chapter apart from the 5 marker. So in 3 marker, anything related to menstrual cycles, spermatogenesis and placenta have more chances to come because these are the topics which are included in your MQP. But again, anything extra can also be coming in, but be prepared for these concepts first. This is your first priority, then comes anything else. You get it? So in menstrual cycle, the hormones which are involved in each phase, what are the unique things that are going to happen, what is the thing that is going to be formed, all of it, you should know each and every detail of menstrual cycle. Any question can be popping up. Spermatogenesis, for example, I've given you a question over here, study the following chart, name the hormones involved at each stage, or probably you can get direct questions on pituitary and testicular hormones involved in spermatogenesis. So if you just go study spermatogenesis and just go to the examination hall. Okay, from here to here the cells divide and that's how spermatids or spermatozoa are formed. No, that's not enough. Anything related to this, hormones, okay, this, related to the process of spermatogenesis are also super important. You get it? So please know about that. That is what I'm trying to say. Similarly, placenta, the uses of it, the hormones released. Apart from that, how is placenta being formed? There is chorionic villi. What is chorionic villi? Please know that. So these are the questions that I've already discussed in the 2 and 3 marker session. 2 and 3 marker session. These are new questions out there. Okay, I'll be discussing them in the live. Apart from that, if you have seen the video on fixed 2 and 3 markers, separately I did a video on fixed 2 and 3 markers. In that PDF also, I've given you the answer for this. Okay, with every video that is being released, we will be republishing all the important videos and the PDFs out there. So you can just get a hold of it. Yes, so for that you have to join the WhatsApp channel. I hope you have joined it. If not, just scroll down in the description box. You will be able to see to get the lecture notes and PDF. Click on this link. So basically that's a WhatsApp channel. At any point when you want to move out, you can move out. It's just for you to get the PDFs and all the important updates out there. Okay? Yes, I'm very active out there. I type the messages for my subject. So yes. Okay? Yes. Now human reproduction 5 marker, my dear children, these are the questions. But over here, a diagram based short question can be coming from this particular chapter like this. So please make sure you know about all the diagram, you know how to make it. The schematic representation of spermatogenesis or basically oogenesis can also come. These are the diagram based questions that have high chances for coming in the 5 marker category from this particular chapter. Okay, apart from that, I have mentioned all the other questions as well. The questions marked like this are the newer questions which have not been discussed. We'll be discussing it more towards the live. Yes? Yeah. If you have a doubt, you can solve it from your end. If you have a doubt, you can please confront me. Now, reproductive health, these are the topics out there. Now, more chances for infertility and art-related concepts. That is in 2 marker category. Infertility, as in how infertility is caused and things like that. Art as an assisted reproductive technology. So, these are the questions which are there that you need to practice. In 3 marker, more chances for STI, sexually transmitted infections, and MTPs, medical termination of pregnancy. When was it legalized and when it is safe to do and things like that. STIs are also known as venereal disease. What are the symptoms of venereal disease and things like that can be asked. Listen, this chapter gives you only 2 and 3 marker and apart from it, MCQs. So you have to grab those 5 marks by solving all these questions out there. You get it? Yes. You will do it, right? Yes, you will. You are my children. Now, principles of inheritance and variation over here, you have all the 2 marker questions that can be coming in. Now, chances for linkage and recombination related question. It can be twisted question also. I have given you certain question examples. Make sure you look at them. Pedigree icon related questions can also be coming in. These 2 are the things which have high chances to come in. Make sure you practice them really well. Not just these 2 things. I'm talking about these 2 topics. Now in pedigree icons, you have many more icons. Make sure you know about all of it. Okay? And know how to make a pedigree chart. You'll see in the 5 marker session. So yeah. In the 5 marker session, there are 2 questions that are going to come from this particular chapter, that is principles of inheritance and variation, 2 5 markers. So basically get 10 marks from this chapter only if at all you're able to do it. One question, one of the 5 marker question can be a schematic representation of either of these, that is 1 gene inheritance, 2 gene inheritance, that is monohybrid, dihybrid cross, or incomplete dominance. Next question can be related to genetic disorders, my dear children. genetic disorder related question. It can be Mendelian disorder, it can be chromosomal disorder, anything. They can give you symptoms, they can ask you. So please know about that. So over here, my dear children, when I say schematic representation related question, this can come. Or if you've seen, this is a dihybrid cross based question. This can also be coming in. So when you provide this, you will be automatically able to do this question also. You get it? When you provide this, you will be able to solve this question also. Similarly, genetic disorder question, something straight like this can be coming in. This is given in a MQP. Okay, apart from that, a question like this can also be coming in. This is also related to genetic disorder, or probably a question related to this, that is formation of pedigree chart, also related to genetic disorder only. So please make sure you practice these HOTS questions are there. There are high chances for them to come in. Now, molecular basis of inheritance in 2-marker category, these are the questions which are there. Structure of nucleosome has been asked in 2 MQPs. There are chances for some diagram based question. One diagram based question, there are chances for it to come. It can be nucleosome or probably transcription unit, or probably there can be something else. For example, HGP related questions, Human Genome Project related questions are there. So there are multiple questions that I have put over here. Make sure you solve it. Apart from these 2 topics also, there are questions being covered. Solve it up. Do not just be lazy to try it up. Make sure you do. What if you make the best use of it and get the best marks? There are high chances for that, right? So make sure you take the utilization for that. And now in 5-marker category, my dear children, these are the questions which are there. Lac operon related question could be coming in. Now, transcription and RNA polymerases, as in different, in prokaryotes, transcription is different, in eukaryotes, transcription is different. Please know the difference between that. RNA polymerases, different RNA polymerases are there. They transcribe different things. There are 3 polymerases. Please know what are they going to transcribe as well. Now, search for DNA. Again, there are 2 5-marker questions that can be coming from here. It can be either this, or probably this or this. So make sure you know how to balance it. Search for DNA in your MQP. Griffith experiment has been asked, but it has been asked from so long, so probably Hershey and Chase can be asked in your paper. And Hershey and Chase is the final proof, so they can ask you in different format. They explain the experiment, which is the final proof for understanding the DNA's genetic material. So it was Hershey and Chase experiment. Now in that also you have subparts, so you should know all the small details which are there. Now genetic code related questions are also being put down over here. When I'm saying you have to practice these questions, I literally mean you have to practice each and every question. And at some point you might feel that I'm so done. No, do not stop there. Complete it as much as you can. Okay? And with experience, I'm telling you that. Now, going ahead, we have these questions as well. So I've just solved the question over here. This looks very big, right? But it's very small answers that they have. Only if you know the process, you will be able to answer it. So please solve these questions. I've given you other answers. What else do you need? Now, human health and diseases, you get 3-marker and a 5-marker. In 3-marker, any infectious disease, their causative organism, their symptoms can be asked. This has a high chance because in 3 MQPs, there are these type of questions out there. Now, infectious diseases in the first part of the chapter, typhoid, malaria, all these things have been asked in different ways, different combinations. Until ringworm disease, you should know everything, their causative agents, their symptoms, everything. Now, apart from that, yes, a diagram-based question of an antibody molecule can be coming in. Not very sure about it, but yes, be sure you practice it. Now, over here, the life cycle. In the 5-marker session, the life cycle of the missing data of either of them. HIV virus life cycle, they can give you missing data, or probably it can be Plasmodium also, malaria's life cycle. So make sure these 2 life cycles are super clear to you. Know the diagram of it, the diagram that is given in your NCERT, super important. Apart from it, you have cancer and immunity-related questions that can have more chances to coming in. Cancer, every part of cancer is important. Apart from that, immunity-related questions as an innate and acquired immunity. Okay. Difference between that, or passive and active immunity. Difference between that, the examples for that, super important. Yes? Now, so as I said, over here, you can get questions like this also. Diseases, their causative agents, even though we discussed in 3-marker category, this is a 5-marker question that have been asked in your question bank. So, when you study for that, you basically study for this also, right? You would be able to answer this also. So, that's all. Now, in evolution, only 2 and 3 marker questions are going to come in. These are the questions which are there. My dear children, in 2 marker category, analogous and homologous organs, all the examples, please know about that. And evolution of plants is something that you should be not missing. They have asked you a question based on that. I do not know how come, but yes, it's there. It's a diagram that is being put in your NCERT. Make sure you know about the diagram, in which era, what organisms are dominant, and things like that. Yeah, apart from it, the questions which have more chances to come, I have put it over here. These questions are new. We have not solved it before. We will be solving in the live, or I have given you the answer in the 2-3 marker fixed video. Now, 2-3 marker fixed video, I have made a separate video on just the 2-3 marker questions are there. Those of you struggling with the 2 and 3 marker questions, there in the PDF where I have given you the questions, I have given you the answers also for this. Now, 3 marker, there's a high chance for diagram of SL Miller experiment to come in, or probably anything related to the experiment can be coming in. So make sure you practice that. Apart from it, adaptive radiation seen in the case of placental mammals and Australian marsupials. These are also super important. So know about that also. Okay, so go through it for sure. Apart from it, the newer concepts which are there, I put it over here. Hardy-Weinberg principle, everything is there. Yeah, but yeah, this should be a first priority, then everything else. Now, microbes in human welfare, again only 2 and 3 marker questions come from here. So in 2 marker category, you can expect anything related to the sewage treatment. Each and every step of it, each and every tank which is being used is important. The name of the tank, what is happening in the tank, where is activated sludge being formed, what is floc, what is BOD, everything is important. Now, biofertilizer related questions also have more chances to come. That is mycorrhiza as a biofertilizer, or probably cyanobacteria as biofertilizer, or probably bacteria as biofertilizer, anything. But again, there's a broader concept in this particular chapter. And remember, in microbes in human welfare, you get only 2 and 3 marker, no 5 marker questions. So you have to practice them. You have to practice the other options also by default. Okay, now in 3 marker category, bioactive agents have more chances to come, because in 3 MQPs they have been asked. But do not be strict to do not just stick to bioactive agents. Okay, know about it. That is, what are the fungal organism that produces bioactive agents, what are the bacteria which is involved in bioactive agents. Okay, for example, you know, cyclosporin A and statin is produced by fungus, whereas streptokinase is produced by bacteria. Apart from it, the other industrial products which are being produced, citric acid, acetic acid, who produces it, all of it, the enzymes, lipases, pectinases, why is it used, know that. So yes, I have included most of the other type of questions also over here, which you need to practice. Do not mess it up. I am telling you again, the ones that are colored differently in 2-3 marker, I have given you the answer in the 2-3 marker fixed questions. Now, biotechnology principle and processes, only 5 marker questions is going to come from this particular chapter. Make sure you study the PBR 322 very nicely. You know what is transformance, you know what is recombinance, everything. Other than that, the tools in the process involved here have more chances. As in tools, as in what are the vectors that you use for the plants, what are the vectors that you use for animals, what is gene gun, what is the vectorless methods that you use, what are the other enzymes that you use, very, very important. Okay, and all the steps which are there. They can ask you, what is this process known as? So you should be able to answer it, right? Yeah. Now the 5 marker new questions that I've added up, I have not discussed before. So yes, you can try to solve it by yourself or be in the live as well. Also, that is one very important thing that I want to tell you. And yeah. Now, biotechnology and its application, I was not able to deduce what could be coming in, because in 3 MQPs, there are 3 different topics. One is transgenic animals in one MQP. In another MQP, it is genetically modified plants. The third one, it is biotechnology in the field of medicine. So, when you solve this, you basically cover insulin and everything. You are covering the whole chapter. And if you cover it well, you get the 5 marks, straight 5 marks, right? So, yeah, these are the different questions that you need to practice. Please make sure you do. Now, again, this looks big, it is not. Please understand, it's only one-liner answer that you have. Make sure you solve it. Now, you have organisms and population. In here also, I was not able to deduce what could be coming in. So, population interaction, that is biological interaction related questions can be asked, and something like this can also be asked. So, make sure you know how to fill in the blanks out there. And this was being asked in MQP, what is mutualism and everything. Apart from that, there's a newer question that I have put over here, such that you know the practice of population growth. So, yes. And finally, population density. This is also MQP. This is also MQP. And something like this also came in MQP, not this. This is a different question that I put. Something like this came. So, please know how to solve this question. It is very important. Okay. Now, ecosystem, only 3 marker question is going to come, and that is this. Those are this. Decomposition related questions could be coming in. Now each and every part of decomposition again is important, which is a process where decomposers are not being utilized, humidification. Or what are the conditions for decomposition, in which condition it does not happen, what are the decomposers in pond ecosystem, everything you should know. Okay, apart from it, so this has a high chance to come. Apart from it, one more thing has a high chance to come. Either this or this could be coming in, and that is formation of ecological pyramids. You have to make an ecological pyramid and show it can be inverted, it can be upright, so you should know the exceptions. Okay, for example, in aquatic ecosystem of biomass, you get inverted pyramid. Now, what are the organisms which are there? You have to make a pyramid and check what are the units that are going to put 100 gram or whatever thing is there. Please know that. Okay, very, very important. Please practice it. You can get question based on this or this. Now, biodiversity and conservation, only 2 marker questions. So basically these are the type of questions that could be coming in. Now conservation methods, this is last chapter, okay, by the way. Conservation methods as in situ, ex situ conservation. In ex situ, what is the advanced method? What are biodiversity hotspot? What are the examples for sacred groves? Everything is important in conservation method. Now ways for loss of biodiversity, as in basically the evil quartet out there, each part of the evil quartet can be asked separately also. So there are 2 things which are high chances. Apart from it also, you have other questions. Make sure you go through it. Yeah? Yes. And as I said, I have solved most of the questions in the 2 and 3 marker category. In the 5 marker category, 2 and 3 marker, basically it just requires 2 hours 30 minutes. If you want to watch it, you can watch it completely for finding the solution. Or if you want to attend the live, in the live I'll be solving the questions are there, you can do so. But if you feel that I do not want to attend the live, ma'am, I have a restricted time, I want to study like that, go ahead with these videos. Again, I'll be pinning it down. This is a 5 marker video. The duration is long because it's 5 marker obviously, and I've covered all of it, which is important. And you will be finding some new questions over here. You do not need to watch this video. Just look for the answers out there. You can just scroll down. This was a live video that I did. It's not recorded. It was a live video that I did. So you can just scroll and check for the answers for any question that you feel is new. Okay. Most of it is being covered in these 2 videos itself. I'm telling you again, if you feel that you're more comfortable in live, because in live you get more chance to interact with me, right? So that is also fine. But if you feel that you're strictly going to be recorded videos, please go refer that. And if you have not subscribed to the channel yet, subscribe to it right now. Well, my dear children, one thing before I talk about this is that do not leave halfway. Do not give up halfway. You have to complete it. That should be your motto. Okay, that should be your motto. And yes, those of you who are preparing for KCET, there is an offer going on for all the courses. KC30, which is an online crash course, you have it available for ₹2499. You have Lakshya KCET offline crash course with a 3000 off as of now, and ABHYAS test series for ₹999. So it's the least it could go, lowest it could go. Make sure you take it up if you're someone who is planning to go ahead with KCET examination. Now that's the only thing that I had to tell you. Wishing you all the best. Please make sure you take care of yourselves. I am here, and yes, I'll be always there. Bye bye. Lots and lots of love from Jasmine ma'am.